All right, today we're diving into a patented method that could seriously change how we look at medical data for cancer detection. We're talking about a move away from simple test results to something way more powerful, a quantifiable risk.
And that really gets to the heart of the matter, right? For years, diagnostics has been a pretty binary game. You either have a marker for a disease or you don't. But what if we could add a real layer of statistical depth to that answer?
And this slide lays it out perfectly. The old way gives you a simple positive or negative. This new method, though, which is detailed in U.S. Patent 11,733,249, completely transforms that. It gives a precise quantitative risk, telling a patient they have a 13.4 times increased likelihood of having cancer compared to everybody else. That is a total game changer for making clinical decisions.
So, here's the plan for this breakdown. We'll start with the big problem this tech is trying to solve. Then we'll get into how it goes from a simple score to a real risk factor. After that, we'll deconstruct the algorithm itself, look at the data that backs it up, and finally, wrap up with what this all means in the clinic.
Okay, first up, the diagnostic challenge. So why did we even need something new? Well, it all boils down to the pretty significant limitations of our current screening tools.
You know, the core issues here are pretty stark. Those binary yes or no results, they just don't give you the full picture. A positive result is so often a false positive, and that leads to incredible anxiety for patients and a ton of costly, sometimes invasive follow-up procedures. And that specific data point from the patent, wow, a staggering 96.4% false positive rate for low-dose CT scans in lung cancer screening. Just think about that number. That is a massive burden on people and on the entire healthcare system.
And this leads us to the real conceptual leap of this invention, that shift from just a raw score to a risk you can actually quantify.
So here's the key takeaway. The technology isn't just flagging the presence of certain proteins or antibodies in your blood. It's taking the levels of those biomarkers, crunching the numbers, and translating all that data into a statistical reality. You know, how much more likely are you to have cancer compared to a baseline? It's all about putting that raw data into a meaningful context. All right, now for the part I know a lot of you have been waiting for, the algorithm itself. How does this whole thing actually work? Let's get into the nitty-gritty.
First things first, the foundation. The whole process kicks off with a multiplex immunoassay. Just think of this as a super efficient lab test. From just one single blood sample, it can measure the levels of a whole panel of different biomarkers, all at the same time. This is the raw data engine that powers the entire system. The patented process really unfolds in 4 key steps. First, the sample gets analyzed with that multiplex assay. Second, and this step is absolutely critical, the raw data gets normalized. This cleans everything up and makes sure values are comparable from test to test. Third, all those normalized values get mathematically combined into one single number, the master composite score. And then finally, in step 4, that score gets stratified into a clear risk level. And boom, this is where the magic happens. This risk categorization table, pulled right from the patent, is that 4th step in action. The algorithm's output, that master composite score, isn't just some random number. If your score lands over 20, for instance, the algorithm translates that to a 13.4 times increased likelihood of having lung cancer. This table is basically the Rosetta Stone that turns all that complex data into a clear, clinically meaningful insight.
Okay, so an elegant algorithm looks great on paper, but does it actually work in practice? Let's dig into the validation data that's presented in the patent.
For anyone working in diagnostics, you know these curves are the gold standard. We're looking at receiver operating characteristic, or ROC curves. They're all about measuring how well a test can tell the difference between 2 groups, in this case, people with cancer and people without. The area under the curve, or AUC, gives us that single performance score. Remember, 0.5 is basically a coin toss and 1.0 is perfect. So what we see here is an AUC of 0.76 in the initial development set and a really consistent 0.73 in a separate validation group. That tells you the model is robust, it's not just overfitted to its original data. Okay, let's zoom in on the performance for those high-risk folks. Now, the sensitivity, its ability to correctly catch those with cancer, is modest in these top tiers. But, and this is a big but, look at the specificity. For the highest risk group, it's 99%. That means it is incredibly good at correctly identifying people who do not have the disease. That results in only 10 false positives out of almost 1,000 at-risk people. This is a direct shot at that massive false positive problem we saw earlier with CT scans.
And we're back to this number.
Because honestly, this is the clinical payoff right here, 13.4x. A score above 20 doesn't just mean you're at high risk, it means a patient has a 13.4 times greater likelihood of having lung cancer. This is the kind of specific, actionable information that lets a doctor guide the next steps with a really high degree of confidence. So let's bring this all back to where it matters most, the clinic. What is the real-world impact of a technology like this for doctors and for patients?
While the impact is pretty huge and it hits on a few different levels. First, it helps doctors refine their screening protocols, triaging patients so that only those with a statistically huge risk get sent for more invasive tests like biopsies. It gives them a much clearer, data-driven starting point for conversations with patients about what's next. And of course, with that super high specificity, you're just slashing the number of false positives and all the anxiety that comes with them. But really, the bottom line is this. It moves beyond just raw data and gives doctors truly actionable insights to guide patient care.
And that really leaves us with a final, bigger question, doesn't it? This patent isn't just about one test. It really represents a whole new way of thinking. As we get better and better at collecting all this complex biological data, the real frontier is interpretation. So the question is, how will powerful quantitative tools like this reshape the very future of preventative medicine? It is an absolutely fascinating time to be at this intersection of data science and oncology.